Welcome back, class. So we've talked about the dual nature of our specific immune response. We've talked about our different classes of antibodies, but let's also talk about our different types of T cells. So one very important kind of T cell is our cytotoxic T lymphocytes. Sometimes they're just called cytotoxic lymphocytes. Sometimes they are abbreviated CTL, and sometimes they're abbreviated T with a small c. All of those mean the same kind of cell. These are really good at destroying our own cells, the cells of a human body that are either abnormal or infected, because we do not want those abnormal cells to spread, and we do not want infected cells to spread an infection, so we destroy those cells. These are identified because they have CD8, that is a protein on the surface of their cell. When they interact with a cell that's abnormal or infected, they will release a chemical called perforin, and they will release a chemical called granzyme. So perforin are going to create pores in the abnormal cell, which will kill the cell, just break it down. Granzymes will stimulate apoptosis. Apoptosis is a way of killing a cell that doesn't just make it explode and spread viruses everywhere. And apoptosis, this is also frequently called programmed cell death. The cell is going to very nicely pack itself up into little blebs. It'll just bulge into these little bubbles and break itself down. It's nice and neat and clean, so you don't have the viruses spreading throughout the body. So it contains the infection and helps get rid of the infection. There's also a class of T cells called helper T cells. Helper T cells have a marker called CD4 on them. So this is a different kind of protein, and they are not going to be fighting with infected cells. Instead, they're going to learn to recognize infections and activate B cells. So a CD4 positive T helper cell will be able to recognize when an infection is flowing through the body. Then it'll interact with the appropriate B cell. Then the B cell will start making the antibodies to kill the infection. There are also suppressor T cells. Suppressor T cells are important because they combat autoimmunity. We do not want our body to make T cells that would destroy our own body, because that would mean we wouldn't have our functioning body cells. So suppressor T cells are there to kill off any parts of your immune system that would fight against your own body. We also have B cells that make antibodies. When B cells are activated, they become plasma cells. And we have natural killer cells, which are lymphocytes. They are white blood cells, although they are non-specific. They are sometimes referred to as the police of your immune system because they do a surveillance. They kind of travel through your body, just checking out cells, see, okay, you're doing fine, I'm wandering on, okay, you're not so fine. When it finds something that's abnormal, it's going to destroy that abnormal cell. So let's talk about T regulatory cells a little bit. These are a subset of CD4 positive cells. That means that they have a CD4 marker on them, but they also have CD25 on them. So they have two different CD markers on their surface. Their functions are first to suppress T cells against yourself. So if your body starts making T cells that will kill your own cells, T regulatory cells should help take care of them. They also protect our intestinal bacteria that we need for digestion. So our good microbes that we want, T regulatory cells will make sure that we don't start an immune response against them. And they also may help protect a developing fetus, increasing research that they are involved in protecting the developing fetus, who will not have the same, may not have the same type of markers on the surface of their cells that we don't want the mother's immune system to attack that developing fetus. Cytotoxic T cells, let's talk about how they work. So here is a cell, and this poor little cell is now infected with a virus. This cell, all cells of your body have what's called MHC class 1 on them. And MHC class 1 is constantly grabbing anything that's going on inside the cell and displaying it outside of the cell. So if this cell is perfectly healthy, doing what it should be, it'll display little proteins and markers on its surface that say, look at all these perfectly healthy, normal things I'm doing. This cell, though, because it has been infected with a virus, it's going to just start displaying virus pieces on its surface. So when that virus is multiplying, it's all over inside the cell. These MHC class 1 are going to start displaying pieces of virus. When it is displaying pieces of virus, it's basically waving a flag around that says, I'm infected, hey yo, come get me, come kill me. So your cytotoxic T lymphocyte comes along, says, okay, I'm just checking out your MHC class 1. Oh, hang on, I can bind to this. I recognize this as a pathogen on here. When that cytotoxic T lymphocyte is able to bind to that, it's very specific. It's only going to be specific for that one type of virus, but it says, I found my virus. It gets so excited, it can't help itself. It just starts leaking all of these things like perforin and granzymes that will kill that infected target cell. So our MHC. MHC stands for Major Histocompatibility Complex. MHC class 1 is what we just saw. MHC class 1 proteins are that red flag that are constantly waving around. This is what's going on in my cell. When the cell is infected, it is going to start waving those around. Say, hi, I'm infected, I'm abnormal, you better kill me before something goes on. So cytotoxic T lymphocytes that have CD8, that CD8 is what will bind to that MHC class 1. So this does not involve antibodies, and at the end, the infected cell is going to be lysed. That's MHC class 1. There's a very different kind of MHC called MHC class 2. MHC class 2 does a completely different thing. So please be able to describe to me what MHC class 1 is doing, what MHC class 2 is doing, what cells respond to MHC class 1, will antibodies be produced, and what's going to happen to the cell with that MHC. So MHC class 1 says I'm infected, so the cell is killed by a cytotoxic T lymphocyte. MHC class 2 is going to be found on cells that are called antigen-presenting cells. These are cells that roam around your body looking for infection. And when they get so excited when they find the infection, and they'll perform phagocytosis. So these are going to be things like dendritic cells or macrophages or even sometimes B cells. So these are cells that are going to go around when they find an infection, they do phagocytosis. And when they do phagocytosis, they keep little bits of that cell, little bits of the antigen, and wave it around saying, look what I found. I found this antigen. This is around your body. Go look for it. Go find it. Anywhere you see this, go kill it. So T helper cells are going to respond to MHC class 2. This is the CD4 marker that they have that interacts with MHC class 2. When the T helper cells interact with it, they don't kill that cell because that cell with the MHC class 2 is not infected. That cell with the MHC class 2 is finding the infection. So we don't want to kill those cells. So those cells are going to stay alive. The T helper cells are going to go activate a B cell to make antibodies, and the antibodies are going to go find the antigen and get rid of it. Okay? So MHC class 1 is on a sick cell, and that cell is going to be killed. MHC class 2 is on an immune cell. That cell is not going to be killed. It's going to help find other infections. Okay, I hope that makes sense to you. If it didn't, let's come talk about it. Read your textbook. There's some accompanying videos that help explain that, because this is pretty critical if that cell is dying or not. Antigen-presenting cells will be cells that are not lymphocytes, things like macrophages and dendritic cells that are going to do that antigen presentation. They have the MHC class 2 on their surface. So after they phagocytose a pathogen, they display part of that pathogen, and so these are going to be found in your organs, your tissues, your fluids, your M cells. When they display that part of the pathogen, it activates your T helper cells to go find that infected cell and get rid of the infection. Antigen presenting cells will display the antigen to your T helper cells and activate them to make antibodies. Oh my gosh. Let me try this again. Antigen presenting cells will present the antigen to T helper cells, which will activate your B cells to make antibodies to go find the infection. Okay, finally got it right that time. So now you can tell me what type of T cell is generally involved when a B cell reacts with an antigen and produces antibodies against the antigen. We're going to come back and talk about B cells and humoral immunity, how B cells get activated and what they're going to do in our next video. I'll see you later. Bye.